So today we are glad to have Hussein YASSINE giving us a seminar. Hussein is professor at Keck School of Medicine at University of Southern California. He leads their translational research lab on lipids, nutrition, and Alzheimer's disease with a specific focus on APOE. He is also a clinician. He leads different large projects on Alzheimer's disease, on lipids and Alzheimer's disease, and also vascular risk factors. Between 2019 and 2021, we co-led together the professional interest area on nutrition and metabolic disorders from the Alzheimer's Association, and this is where we met. So I'm really happy that Hussein could visit us in Bordeaux during his European tour. So thank you very much, and the floor is yours, Hussein.
Thank you, thank you. Thank you, Cecilia, for this invitation. I'm really happy to be here. And I know a little bit of French, so I can get by, but this talk is going to be in English. I don't have any relevant disclosures for this talk. I'm going to try to end it 10 minutes early so we have a chance to discuss, talk, ask questions. I'm going to try to convince you in the next 50 minutes that knowing the APOE genotype is important and possibly can change how we treat and manage patients who are at increased genetic risk of late onset Alzheimer's disease. The talk is going to be broken into a section where we look at modifiable risk factors, APOE4 and dementia, omega-3 metabolism in the brain before dementia, omega-3 metabolism in the brain after dementia, and then finally a few slides on the pros and cons of genetic testing to figure out if you carry E4 or not. So basically, the argument that I'm going to pose is that the effect of E4 on the brain can be modified. And if we understand how APOE4 changes metabolism, whether it's lipid metabolism, glucose metabolism, fatty acid metabolism, we can possibly modify recommendations or treatments to protect the APOE4 brain. A lot of this introduction is already published, and you can find them in references, but I hope that we can get something or a message that we can change how APOE4 affects the brain. So why APOE4? APOE4 is a common gene variant. The actual prevalence of E4 varies by country. For example, in Nigeria, up to 50% of people have one copy of APOE4. In certain populations like Latin America, where that ratio can go as low as 10%. In Europe, it's about between 10 and 25%, depending on the country. But more importantly, the association of APOE4 differs by ethnicity. Not all E4 carriers have the same risk of getting dementia. In white people, especially in Japan or Asian countries, E4's risk is incredibly high. One copy can be 4 up to 6-fold increase. In other, it's 1-fold or less. But if you have one copy of E4, the reported average odds ratio is 4 times. And if you have 2 copies, your odds ratio of getting dementia can be up to 10 or 12 times. Giving you a little bit more insights, if you live to become 80 years old, and that's a survival advantage, and you are an APOE4 homozygote, you have a 50% chance of getting dementia. So it's not all doom and gloom. There's another 50% who will never get dementia, who have 2 copies, but you have to survive to 80 to be able to get that statistics. I just want to take you just a minute to look at this table and the variation in the odds ratio of dementia by region. And you could see that in Africa, the odds ratio is significantly lower than Europe, North America, and so forth. So there's a big variation in how APOE4 increases the risk of dementia. But more interestingly, this is an old study back in 2001 where they looked at Yoruba Nigerians who live in Nigeria and looked at their estimate of getting dementia and compared them to Africans who live in Indiana in the United States. And what you could see in the slide that there's a big difference between an African in Nigeria getting dementia, much lower relative risk, versus an African American who now lives in the US. And to us, that means something is happening to a migration of E4 into developed countries where there's a mismatch between genes and the risk of dementia. So I'm going to walk you through the next 30 minutes or so on this hypothetical model of what happens to cognition across the lifespan in an APOE4 carrier. This data is not purely theoretical. In fact, there are many longitudinal studies which have modeled this, one of it by Richard Caselli from Banner in Phoenix. But what you could see is approximately in the first 55 years of life, E4 carriers don't show strong changes on cognition. Now, some of you will prove me wrong and develop very new tools and find subtle changes, but at this time and place, the common tests that we do for studies do not convincingly show that the E4 brain is declining in the first 55 years of life. Between 55 and 70, we start to see cognitive decline more in homozygotes than heterozygotes. And this phase is often described by clinical researchers as a prodromal AD or pre-AD, where somebody has cognitive decline, but they're not demented yet. They don't have a diagnosis of dementia. And then you've got the diagnosis of dementia where now cognition and function are both affected, and it's a rapid phase where there's accelerated cognitive decline. So I'm going to try to convince you that in stage 1, we have changes in the brain that we can see, and they include changes in amyloid, a relationship with physical activity, and importance of omega-3 intake. I'm going to show you data on bioenergetic stress at the blood-brain barrier in the preclinical phase, just before people get dementia. And then I'm going to argue that there is a switch to polyunsaturated fat oxidation and catabolism once dementia ensues. So the next part of my presentation is going to walk you through 3 different phases of what the APOE4 brain is going to go through. So let's start with amyloid. So many studies, and this is just one of them, show that amyloid starts to accumulate early in a homozygote in the 20s, in a heterozygote 30s and 40s. So if you do PET scanning with an amyloid tracer injected to people and see how the amyloid is lighting up using 18F, you could see that the amyloid brain, the APOE4 brain is accumulating amyloid as early as in the 20s if you have 2 copies of E4, and as early as 30s or 40s if you have 1 copy of APOE4. Now, we know that this is complex. Some E4 carriers get more amyloid doses and then other E4 carriers. And in this systematic review from a collaboration between Australia and Spain, they actually identify that younger APOE4 carriers tend to have less amyloid doses if they are more physically active. And some of these studies, and it's a very busy slide, but the point is some of these studies suggest that physically active younger APOE4 carriers are less likely to have dementia later on in life. Of course, there's a lot of heterogeneity, there is inconsistency, some reports show you the exact opposite, but studies with large sample sizes often show a robust association between greater physical activity and better cognition in E4 positive. My lab works on omega-3 metabolism, and I'm going to try to convince you that the same signal can appear in younger APOE4 carriers. For example, in this Finland study, you could see that younger APOE4 carriers between the ages of 40 to 55 who consume more polyunsaturated fat have better brain functions than those E4 carriers who consume less polyunsaturated fat. In the study from Rush, Marie-Claire Morris showed that E4 carriers who consumed more seafood had less AD neuropathology than E4 carriers who did not consume, and this is a brain autopsy study. In a young randomized clinical trial, these people are at the age of maybe between 20s and 40s. 1 gram of DHA per day was associated with improved memory outcomes, in this case reaction time. And in the finger trial, E4 carriers were more likely to respond to an intervention that had both exercise and increased seafood consumption than non-E4 carriers. So DHA, some of you are familiar with this omega-3 fatty acid. It is neuroprotective. and it makes a significant component, although this number might be a little bit inflated, but it makes a significant component of the omega-3 in the brain, has anti-amyloid properties, has anti-inflammatory properties. And we have published many years ago that being in the lowest quartile of DHA associates with more amyloidosis by PET-PIB scan in the brain. So I've been interested in studying how omega-3 get into the brain. And my mentor at the time, Stanley Rappaport, developed modeling methods using tracers that can tell you how much DHA can go in and out of the brain. One of the findings that really struck with me is that when you inject 11-C-DHA and study how much it takes to disappear from the brain, you can do some mathematical modeling. And in this study, the model suggested that the half-life of DHA in the brain can be up to 2.5 years. Why is this important? It's important because this means if you do a small trial with 6 months, you're not going to sufficiently remodel the neuronal membrane DHA. Now, the turnover in synapses is going to be faster than non-synaptic, but in general, it's a slow turnover. That also means it's good news if somebody stops eating omega-3s for 2 to 3 years. The brain is not going to shrink rapidly. The brain has time to hold on to its own omega-3s. So I was interested in studying the effect of omega-3s and APOE4, and in this study, we recruited individuals who don't eat enough seafood, and then injected them with 11-C-DHA, and then monitored the uptake, or K-star, of DHA into the brain. What we found is that younger individuals, 33 mean age, had greater uptake of DHA into the brain compared to non-carriers. To us, that meant that the APOE4 brain is hungry for omega-3s. It's sucking more of it from the plasma into the brain. What are the implications? Well, if you take an APOE4 carrier and deprive them from omega-3s, you're going to induce some form of stress. So we use this tool, but we also recognize that it's a problematic tool because 11-C compounds have a very short half-life. So over a period of 2 to 3 years, Marlon Duro, a talented postdoc, a chemist, was able to make 18 FDHA, and then we validated the signal. We injected aracholine. It's a small molecule that can increase DHA uptake, and found out that by this modeling in mice, we can see DHA get into the brain. We plan to use this modality now to study disease models, E4, non-E4, poor diet, other environmental risk factors, and see what the effects on DHA uptake are. So we talked about phase 1, which is the normal cognitive phase. Let me now shift gear to phase 2, which is bioenergetic stress, or why the blood-brain barrier is showing signs of stress and unable to keep up with having this polymorphism, or polymorphic APOE. So take a look at this slide, and what you could see is that if you examine the blood-brain barrier by an electron microscope, you see a predominance of mitochondria lining up the actual endothelial cells, pericytes, and the cells that make up the blood-brain barrier. So the brain is actively consuming a lot of energy to pump nutrients across into the brain itself. There is an active mechanism, and we recently worked with Cecilia to publish a review on bioenergetic failure in Alzheimer and dementia. But in a nutshell, we show that many transporters, whether it's going to be the DHA transporter, MFSD2 alpha, the glucose transporter, GLUT1, the ketone transporter, MCT, they're all energy-demanding activities that are susceptible to a failed blood-brain barrier. Indeed, when you actually do fancy MRIs that we're starting to do now at USC, we can compute a new variable called KW, or the water transport coefficient. You don't have to inject any contrast. You just use a regular 3T MRI with fancy sequencing that can compute KW, and you could see that in the first panel, APOE3, APOE3, and APOE3-4 and 4-4, there is a lower KW in those who carry the APOE4 that associates with more amyloid plaque deposition, which is the lowest panel where you can see more red color. And there's a correlation. Danny Wang from USC led the second study in Alzheimer and Dementia 2021, where you see a correlation between CSF beta 42 and KW. The higher CSF beta 42, which in our world means less amyloid deposition, associates with a higher KW. So as the brain is accumulating more A-beta, the function of the BBB is getting less. look at glucose now, and you could see that in cognitively normal people, and this is a relatively large study from the Mayo Clinic, between the ages of 60 or 55 to 70, this bioenergetic stress period, you see that there's signs of glucose hypometabolism, increases by APOE4, and having more amyloid is a second hit that makes that worse. Our own lab ran a randomized clinical trial that we call the DHA pilot trial, and in that trial we recruited 33 individuals over a period of 2 years. This was led by Isabella Cordoba Arianes, and the team found that, it took us about maybe 2 years to find people willing to do a lumbar puncture at baseline and at 6 months, and then 50% had to be E4 positive. They had to not like any fish to be able to participate, so we did questionnaires on their fish intake, and we gave everybody vitamin B12 because we know that there's an interaction between omega-3s and B12, and we did not want low B12 to be a confounding variable. And this is published, so you can see the actual baseline characteristics of this trial. I'm not going to spend much time, but what we found is even in cognitively normal APOE4 carriers, before they have any dementia, between the ages of 55 and 80, there is evidence that E4 positive individuals had less DHA and EPA and CSF compared to E4 negative individuals, telling us that during this period of bioenergetic stress, it's not just glucose, it's DHA and possibly other metabolites that are having a problem getting inside the brain. So I hope to have convinced you that the APOE4 brain might be more dependent on circulating DHA, and that's largely a function of peripheral intake. But still fundamentally the question remains, can DHA supplementation in cognitively normal E4 carriers with limited DHA intake delay the onset of AD? So from 2018 till now, we're actively doing a relatively large single-site clinical trial that we call Prevent E4, where 368 individuals are randomized to 2 grams of DHA versus placebo over 2 years. We started recruitment in 2018. We were supposed to finish in 2021, but COVID didn't like our plans, so we had to pause for approximately 1.5 years because we lost a lot of people who decided during COVID we're not going to be in trials anymore. It's not a priority, so we had this hit, but we were able to pick up and finally finished recruitment in May of 2022. This trial is supposed to be published hopefully by the end of 2024, and it's funded by both NIH and ADDF, and we received no funding from the DHA makers. The primary outcome is going to be, again, CSF DHA changes by genotype. We're going to look at cognition and MRI changes after 2 years of supplementation. And this is an idea of how the flow of that trial is. We had to actually screen over 5,000 individuals, fill out questionnaires for 3,000 individuals to ultimately get 368 in a single site, which was a lot of work, and a huge team had to work excess hours to be able to make that deadline. So this is the baseline findings, and they will be soon reported in the JPAD journal. The mean age is 66. We had 35% representations from Latinos, which was super exciting, but this is a double-edged sword because the underserved population that agreed to participate in the study ended up being the population that we lost the most by follow-up because they have less resources and less likely to be able to continue. But we're excited that we were able to get this representation from minorities. Another question that lingers is 66 too old? Is this really the time to replenish with DHA? Did we miss the boat? We don't know yet, but we're excited to find out what Prevent E4 will show. So let's go now into the third phase of this talk, which is omega-3 metabolism in APOE4 dementia. And this is a phase that I'm defining as a treatment phase. So I want to talk to you right now about how this metabolism of fatty acids is going to change. So I showed you that during bioenergetic stress, there's going to be some glucose hypometabolism, lower DHA delivery into the brain. What I'm trying to convince you now in this next phase of the talk, that the brain, because of bioenergetic failure, or some form of a failure, it's going to start eating itself up. And it does so by oxidizing and catabolizing polyunsaturated fatty acids. We already know from epidemiology studies that older APOE4 carriers are less likely to respond to DHA or to polyunsaturated fatty intake in seafood compared to non-carriers. And this is a paper that was led by Cecilia that we published together a few years ago, showing that in epidemiology studies, E4 carriers tend to less respond to omega-3 intake than non-carriers. But most of these studies had people in their 60s and 70s. In a trial led by Joe Quinn from Oregon State University, he showed that 2 grams of DHA over 2 years was not effective, period. But when they sub-analyzed into E4 positive, E4 negative, those who carried E4 did not respond at all, while those who did not carry E4 had some response. So something is changing now. We talked about early in life where it does something. Now late in life, it seems that PUFAs and supplementation is almost doing nothing, if not the opposite. Is it restricted to PUFAs? Probably not. This also affects other nutrients, and these are 2 systematic reviews that looked at ketone response at clinical trials, and they both showed that if you are in a clinical trial where they give you ketone meals as part of a ketogenic diet or a ketogenic intervention, if you're E4 positive, you're less likely or you will not respond. So it's not just glucose, not just DHA, also the MCT transporters can be affected by this bioenergetic stress at the blood-brain barrier affecting the delivery or metabolism of ketones as well. So we reviewed this in detail, and the switch that, as you can see in this figure here, is that something happens where glucose is no longer enough. It still remains the primary source of metabolism for the brain, but the pathway, anaerobic glycolysis, oxidative phosphorylation, is now changed in the dementia brain, and you start pushing or pulling fatty acids from membranes, and that leads to trouble. And we think that when the brain tries to borrow PUFAs for energy as opposed to synaptic signaling, the price might be neuroinflammation. So my lab has been interested in neuroinflammation for the past at least 5 years, and I'm going to show you a series of studies that we've published. First is a collaboration with Zoe Arvanitakis and David Bennett at Rush, where we took post-mortem brain samples from the inferior frontal lobe of patients which were categorized into E3s or E4s, normal cognitive function versus AD, E3s and E4, and this is a small sample size. But we since got funding from NIH to expand it into a substantially larger study that's actively ongoing. But Brandon Ebright and Isaac Asante were the 2 people that led this mass spec study. So here's what we find. We find that in these, and I skipped this slide, when we look at omega-3 metabolism, we look at a family of eicosanoids. And these are prostaglandins, leukotrienes, and other mediators which are broken down from PUFAs. Some of those are pro-inflammatory, as you can see in red, and some of those are anti-inflammatory that you could see in blue. And I know Sophie works on something similar. So what we found is that APOE3 E4 compared to APOE3 has an accentuated pro-inflammatory and anti-inflammatory signaling fingerprint. So you could see much brighter colors in the 3-4 compared to 3-3. Which means in the brain of the APOE4 carrier, there is a sustained chronic inflammation and anti-inflammation. So you move to a state where there is inflammation and anti-inflammation, and that's an entity that's defined as unresolved chronic inflammation. But the story doesn't end here, because when we broke down those groups into NCI and AD, we found that the E4 and NCI actually had no signs at all. You could see that sliver of column 3-4 is mostly blue in color, dark blue, meaning that you did not turn on this activation in E4 carriers who died without dementia. In contrast, those who died with dementia, so you have E4 and dementia, are those who activated the system. To us, that means E4 is not sufficient. You can die with E4 but no dementia. And 2, maybe E4 is modifiable. So if we can understand what are the fundamental mechanisms that drive E4 into neuroinflammation, that could be a potential significant drug target. So we proposed in 2017 that it is possible that CPLA2 or calcium-dependent phospholipase A2 is hyperactivated in APOE4. At that time, the mechanism or the signaling pathway that activates APOE4 or phospholipase A2, CPLA2, involves an MAPK P38 system, and has been shown that P38 is activated in APOE4 but not CPLA2. So we looked at this pathway and examined MAPK CPLA2 signaling in the setting of APOE4. This was led by Xiaowei Wang, a very talented postdoc in my lab that I would not know what to do if he decided to quit research. So I'm trying to convince him not to quit. We studied a human APOE knock in mice, primary astrocytes, and human brains. And I know that this might be a little bit complicated because this is not public health. But the concept is not that complex to understand. So what we find is that in cells isolated from the mouse brain, E4 has more activation of CPLA2. Cells, these are primary astrocytes. Then when we take the whole mouse brain and run a gel on it, we find that APOE4 has more activation of CPLA2 as well. And then when we actually look at the signaling pathway, we find that the P38 system, which turns on CPLA2, is more activated. And finally, when we took post-mortem tissues and stained it for CPLA2, we found more phosphorylation of CPLA2 in APOE4 dementia compared to APOE4 non-dementia. So what this told us is that, yes, indeed, APOE4 is activating this enzyme, which works on the membrane. And if you saw that slide that I didn't spend much time on, you could see here that we drew that CPLA2 is coming to membranes, getting phosphorylated, getting the fatty acids out of membranes, and by a series of LOX, COX, and CYP enzymes, turning into eicosanoids and participating in a host of neuroinflammatory signaling. We got funding, as I mentioned, to examine a much larger database, and this is a relatively new slide. So we now have about 200 slides from Rush, and we have dug deeper into looking at how this looks like in the postmortem brain, and we found that there is activation of CPLA2 in red in astrocytes, which are stained positive by GFAP around amyloid plaques in AD brains. And this is now getting expanded into a cohort of E4 positive, negative, with dementia, without dementia. Another project that we have been working on is, can we actually image or diagnose neuroinflammation in the living brain? And that is another big challenge. The post-mortem studies are nice, but you cannot use them for somebody who's alive. So how can we actually image neuroinflammation in the AD or APOE4 brain? So my mentor, Stan Rappaport, developed an 11C arachidonic acid PET probe, and he injected it into patients with Alzheimer's disease and found out that 11C arachidonic acid is increased in the brains of Alzheimer's dementia compared to those without dementia. The problem, again, with 11C is that it has a very short half-life, so you have 20 minutes to run with the patient into the scanner before the actual compound disappears. So we, again, spent similar time or an effort to DHA when we were able to make the 18F version of arachidonic acid, validated the signal, and published this in PUFA in 2022. And our next step now is to see if APOE4 mouse models have more arachidonic acid uptake in their brains, ultimately creating a probe that we can move to the clinic and image CPLA2 activation in vivo in APOE4 carriers before they die. So then you might ask me this fundamental question: is phospholipase A2 activation in AD a cause or consequence of neurodegeneration? We don't know. However, I want to show you briefly what past papers have shown. It started in 1997 in a paper published in Nature showing that when you knock out CPLA2 from mice and you ligate their blood vessels in the brain, they get less stroke damage compared to those who are wild type. When you knock out CPLA2 from mice and cross them with an APP AD-like mouse model, you get less cognitive decline. When you actually have brain trauma, you drop something on the head of a mouse. I don't know how IOCO can approve it, but they did. You find that if you actually knock out CPLA2 or inhibit it, you can get less trauma. In a status epilepticus, where you have continuous seizures, if you inhibit CPLA2, you get less neuroinflammation. In spinal cord injury, instead of dropping the heavy weight on the brain, you can drop it on the spinal cord. When you knock out CPLA2, you get less inflammation. And finally, in a model of Parkinson's disease, when you modulate this pathway, you get less neuroinflammation. So do we have any active CPLA2 inhibitors? We don't have great drugs that inhibit CPLA2, but the best one on the market is ASP 14780. So we did the study over the last 2 years, has not been published yet, where we took APOE4 mice, put them in a very poor diet that has no omega-3s from the age of 6 months till the age of 16 or 17 months. And then we did behavioral testing, we sacrificed tissues, we did imaging, and the E4 mice were randomized to either vehicle or CPLA2 inhibitor 14780. And this is a model of chronic inflammation. So what we found is after treatment with ASB at 16 months of age, in mice, 16 months of age is approximately 50 to 60 years in humans. We can increase EPA, we can increase DHA, and we can reverse the EPA-AA ratio and the DHA-AA ratio, which means in this APOE4 mouse model that has very poor diet and really no DHA, where the DHA and EPA were consumed because of chronic neuroinflammation, when we do 2 months treatment with the CPLA2 inhibitor, we can reverse the omega-3s. And then when we looked at the inflammatory and pro-inflammatory pathway, we found out that LXB4, LXA5, which are anti-inflammatory, went up, and PGE2, prostaglandin E2, which is pro-inflammatory, went down after we blocked CPLA2. And then we looked at behavior, and unfortunately the sample size was an N of 5 to 10, was not large enough to see behavioral differences in this model. But when we stained microglia, and microglial inflammation is one of the hallmarks of Alzheimer's, we found out that CPLA2 inhibition successfully decreased microglial activation, as seen in this picture, and stained by IBA1 positive cells. These are markers for microglial activation. Then we said, okay, this is chronic inflammation. Can CPLA2 inhibition also reverse acute inflammation? So we took mice, gave them LPS. This is a lipopolysaccharide. It's an acute way. When you inject it into a mouse model, they get inflamed, and if you inject a large dose, they get septic and can die. So you have to use lower doses, and then you inject them for 5 days, you harvest their brains, and you see all signs of inflammation in the brain. Those mice were randomized into vehicle with LPS versus ASB 14780, which is the CPLA2 inhibitor, plus LPS. And what we found that indeed, after 5 days of LPS, you deplete DHA from the brain, it's consumed. But when you inhibit CPLA2, you preserve DHA in this acute model of inflammation. And that was associated with better markers of, or less markers of neuroinflammation. Now, I talked to you about the blood-brain barrier, so what about the blood-brain barrier? So one of the models for BBB dysfunction is an APOE knockout mouse model, where they have big changes in the BBB. So we took an APOE knockout mice and randomized it at the age of 4 to 6 months to either vehicle versus ASB 14780. And what we found is that CD13, which is a marker of parasites, is starting to increase, meaning that there is healing of the blood-brain barrier when CPLA2 was inhibited. In contrast, moreover, we did DCE, this is dynamic contrast enhancement MRI scans at 7 Tesla in mice. That's a kind of fancy tool and quite difficult to do, which regrettably you only find out after spending $100,000. So after a year of figuring out how to do DCE MRI contrast, we found that K-trans, which is a measure of contrast leaking into the brain, as you can see in the photo, is less in mice treated with the ASB 14780, which is the CPLA2 inhibitors, compared to mice treated with vehicle, which means that inhibiting CPLA2 also started to repair the blood-brain barrier. So in this review, we show that CPLA2 activation is really pan. It involves the neuron, leading to a neuroinflammation and neurodegeneration phenotype with neuronal loss. It involves the astrocyte, where you find GFAP positivity and astrocytosis. It involves the microglia, where you find microgliosis, and involves the parasites and the endothelial cells and the blood-brain barrier. And maybe CPLA2 is a good target. And actively, we're collaborating with Seva Catridge and Anastasia, his postdoc, to discover new CPLA2 inhibitors. So this work started about a year ago. We screened 18 billion small molecules, eventually landed with 127 theoretical molecules which have a drug-like phenotype. They're not known theoretically to be toxic and can cross the BBB. From those 127, we indeed identified 8 compounds which can block CPLA2 inside the test tube, and finally landed with 2 parent compounds which we are testing now in vivo in the brain, and show that preliminary studies not shown here, they can cross the BBB and engage with CPLA2. All right, so let me just try to wrap up so we leave some time for questioning. Is should everyone get an APOE genotype testing? And that is really the fundamental question that I started with. So let me argue that yes, we need to know that you are an APOE4 carrier. So as a clinician, I do run an APOE clinic, and people in the US do a lot of 23andMe. They want to know their genealogy, where they come from. And they get surprised when they get this report and they see that, oh, you're carrying 1 or 2 copies of APOE4, you have an increased risk of dementia. What do they do with this information? They need answers. They need to know if there's anything they can do to stave off the risk of dementia. If you have Disney Plus, like we do, there is a series known as Limitless, and Ken Hemsworth, which I didn't know before my children told me about him. He is some Hollywood guru, which I don't really follow. But Ken Hemsworth, in one of the episodes in Limitless, talks to Peter Attia, who has a very famous podcast in the US, and discovers that he's an E4 homozygote. And that changed this actor's life, because once he found out that he carries 2 copies of APOE4, he actually stopped acting now. And he's dramatically changing how he's living his life, because he doesn't want to get Alzheimer's dementia. So if you knew that you are E4 positive, then you probably would start doing things differently. If you're concerned about the risk of dementia, then you would think about lifestyle modifications. Maybe change your diet at a young, early age, increase your consumption of polyunsaturated fat, becoming more physically active, avoiding trauma. Like the patients I have who have children who are E4 positive, I tell them, don't engage in American football or any contact sport where you may have brain damage. Take care of your diabetes, hypertension, high cholesterol at an early age. Because once you transition into this catabolic phase, these interventions likely may not make a big difference. However, the problem with having APOE genotyping is, 1, you have to defray the cost. It's not free, but it's not that expensive. Right now, you can get genotype for as little as $25. 2, you need to provide counseling to those who get E4 positivity. You just cannot say, oh, you're E4 positive, adieu. It doesn't work. They're going to have psychological trauma. They're going to be, heavily impacted by this knowledge. But I think knowledge is bliss, and knowledge is awareness. If I were E4 positive, which I don't know, I would want to actually start changing my lifestyle as soon as possible. The issue is also in the US, I don't know if France may be different, is that if the insurance finds out that you're E4 positive, they'll find a way to slap you with additional premiums. And Obama released a new law known as Obamacare that prevents insurance companies from doing so, but they're slicky and they can do things that nobody likes. And finally, we still don't have enough research, despite all the work that we've done, to have strong evidence-based recommendations of what to do for APOE4 carriers in terms of lifestyles, drugs, or other things. So the recommendations are good, but still not backed by evidence-based medicine. So I had the fortune to work with Cecilia and many others as part of the Alzheimer Association. We published these 2 beautiful papers. And one of the papers that we published last year concluded that systematic reviews of randomized clinical trials of dietary and nutritional supplements have largely been negative. And I hope that this presentation could help provide some insights why these modifications, and I know Maud is doing outstanding work on longitudinal modeling of this work, to give you some insights why these interventions have to happen as early as possible, because once the brain transitions into the catabolic state, changing the diet or perhaps exercising is far less effective as opposed to starting it very early on. To be able to do so, though, we need new biomarkers that can capture brain health when you're 30, 40, and 50, decades before you get dementia. Also, the table from the paper in Alzheimer and Dementia in 2022 looks at the ratios of metabolites between CSF and plasma. And this is not just DHA, it's not just ketone bodies, it's not just B12, all the other things that get into the brain. The blood-brain barrier appears to affect their transport. So when you get somebody with dementia, it's going to be very hard to get these metabolites into the brain. It's going to be very hard for them to be metabolized efficiently. So I'm going to conclude that maybe diet and physical activity appear to modify the associations of APOE4 with dementia. Those living in Nigeria who are having their indigenous food, foraging, walking 17 to 20,000 steps per day, made the wrong decision by moving to Indiana. In Indiana, they stopped doing the same activities, started eating fast food, and the risk of dementia was substantially higher. I hope that I have convinced you that the nutrient delivery to the brain starts to change during middle life, and is manifested by bioenergetic stress at the blood-brain barrier. and that younger APOE4 carriers, especially those at risk of dementia, may require better fuels. Like you have a fancy car, you need really premium grade oils like omega-3s to keep that brain happy. And if you substitute that with cheap fuels, the brain doesn't like it or starts to getting stressed out, and the price is going to be after the age of 60 when dementia starts to develop. 18F DHA PET scans that we're working on, we hope that we can use this tool to figure out what's happening to omega-3s decades before people get disease. And similarly, 18F arachidonic acid, we're going to be using those in individuals who get neuroinflammation to identify which of APOE4 carriers start to have activation of CPLA2 and possibly be targeted for treatment. So, you know, we are excited about CPLA2. We don't know if it's going to be a viable drug target. Maybe yes, maybe no. We still need to know whether this is a cause or consequence. That can only be proven by inhibiting CPLA2 and finding better outcomes. At this point, this is all association, but at least there's some excitement that keeps us going. And finally, I hope to convince you that yes, knowledge of APOE genotype is important. I think if you knew what your APOE4 genotype is and you started working on modifying your risk factors at an early age, you are way better off than if you did not. Now, the counter argument is, well, everybody should eat healthy. Everybody should exercise. Well, maybe it doesn't work, because in our clinic, when we tell everybody to do so, they don't. But if they knew that they're E4 positive, they will take it more seriously. And hopefully, they could use this knowledge to prevent or delay this disease. So I'm going to conclude by acknowledging the many folks that actually helped us with these studies, including Xiaowei, Isabella, and others, Helena Chu, Lon Schneider from USC, Mike Vranos, who's one of the funders of the CPLA2 drug development, the Alzheimer Association, NIH, and the Alzheimer Drug Discovery, and our collaborators as Drush that provided brain samples. And you can always reach out to me at my email, and I'm happy to address any of your questions. Thank you.